Well, thank you, Jason. Definitely, it's been really nice to be in this lab, and I'm going to talk a little bit more later. Thank you, everyone, for being here. This is really great to see a lot of people here in this summertime. I wasn't expecting this, so I really appreciate it. And also, thank you to the people that are watching on Zoom. So, well, as Jason said, this is the title of my talk, Comparative Phylogeography of 3 Widely Distributed Amazonian Poison Frogs. So, first of all, if we're going to talk about Amazonian poison frogs, we need to talk about the Amazon rainforest. I'm pretty sure you've heard a lot about it, and I'm just going to give you a few facts. So this forest is the largest continuous forest in the world. As you can see, it's a huge forest, and with this magnitude, it contains over half of the planet's remaining rainforest. And it also is a very important ecosystem because it serves as a major source of biodiversity for other neotropical ecosystems, meaning that from this area, a lot of groups have migrated out of it. And I'm pretty sure you've heard about this, but the Amazon is a highly diverse place, and indeed it hosts more species per unit than any other region in the world. So now that we know about the Amazon, we of course need to talk about amphibians in this ecosystem. So the Amazon harbors the highest species richness in amphibians in the world. Some estimates talk about 600 species, but however, with molecular data, we think that there's a large underestimation of that number, and there could be more than 1,000 species. So with this number of species, one way we could think about how we accommodate all of these frogs is by having restricted ranges. And that's something like that. You have a lot of frogs, and all of them have little tiny ranges. And one reason why is that could be due to their low dispersal ability. However, one of the main reasons that explain these restricted ranges, and not only in amphibians, but in a lot of other groups, is the changes in geological and climate dynamism that this region have gone through over the past 10 million years ago. So even though we have these restricted ranges, we still recover some frogs that are capable of distribute all across the Amazon basin. And this is very weird, and that's why I am interested in what's going on with these weird frogs that are able to just be there. So one of my questions is what are the historical processes that have enabled these species to just overcome this dynamism and then still till this time persist as one single species? Okay. So, well, here is my research question actually. So it is, what processes and mechanisms drive colonization histories of Amazonian species with large distributions, and also if those processes and mechanisms are shared among species. So in order to answer this question, of course, I'm going to be talking about poison frogs because we have some widely distributed poison frogs. I'm going to briefly introduce this family. So as you might know, this is a highly diverse family. It has over 200 species. And within this diversity, at least 30% of species have ranges that are in the Amazon rainforest. So, and this family also have this characteristic with more frogs have restricted ranges. However, we have some frogs like this one right here, the Amerega trivitata, that, as you can see in the map, it has a continuous and very large distribution from the west till the east in the Amazon. We also have Amerega haneli, which also, as you can see, has a continuous and very wide distribution. And on the other side, we have this other frog, Ranitomea variabilis, which is quite different to these other two because we see this huge gap in its distribution right here. This is what we call a disjunct distribution. And in order to understand a little bit more about this disjunct distribution, we need to talk about its sister species, Ranitomea amazonica. And the reason why we need to look into these two species is because there has been some taxonomic incongruences regarding the validity of these two species, mainly because these eastern populations right here, right now belong to Ranitomea variabilis. However, in the past, using mitochondrial genes, they were assigned to Ranitomea amazonica. And it was just like recently with one of Jason's students' work who used nuclear DNA, and then she found out that indeed these populations are part of Ranitomea variabilis. So in order to understand what's going on with this species, we need to talk about both. So I'm going to be referring to these two species as Ranitomea variabilis amazonica species complex. But keep in mind that my focal species will always going to be variabilis because it's the one that has the huge distribution or the wide. So these species, even though they have wide distributions, they are quite different in several aspects. I'm not gonna explain all of this, but mainly they have different sizes. We have big frogs, like 5 centimeters, which is huge for a poison frog. And then we have this little tiny that is like up to 2 centimeters. They have differences in altitudinal ranges, reproductive ecology, and where they deposit tadpoles. And I'm going to be talking a little bit more about this later when we get into discuss this species. So in order to answer my questions, I'm going to just give you a brief overview of what I'm going to be talking now. So I'm going to talk about my bioinformatic analysis, the population structure analysis that I perform, phylogenetics, time-calibrated phylogenies, historical phylogeography, comparative phylogeography, and then finally my conclusions. It is important to tell you that except for the population structure analysis, all of the first 4 steps are the building material for my last 2 steps. So I'm going to briefly explain the first 4 points, and then whenever I get to the last part, I'm going to integrate all of my results into one single history so you don't get bored. So for my bioinformatic analysis, I use nuclear and mitochondrial DNA. Oh my gosh. Well, so the data collection, I have a data set that is built from many field seasons across several years. It wasn't like I didn't collect like basically any of these samples, because in order to have a huge data set where you can have cover the distribution range of all of the species, you need a lot of field work. So I work with a bunch of data that was already collected. It was already extracted, the DNA. I went to the field with a few years ago, and then I collected some of the samples, and few of them are in my data set, but the majority are not. So after I get all of my samples, we are working with ultra-conserved elements, which are part of the nuclear DNA. And so what this means, as the word says, they are highly conserved elements that you can find in the genome, in the nuclear genome. However, there are flanking regions that are highly variable because they accumulate mutations. And this is what is very informative for evolutionary relationships. So this is my nuclear data set. And I am also gonna work with mitochondrial DNA. Basically, even though sometimes, like when you send your sample for UCE sequence, if you have tissues that have a lot of mitochondria, like muscle or liver, you can also get some off-target sequences, like mitochondrial DNA. And if you have the right bioinformatic tools, you can just harvest those mitogenomes, and then you can also get a mitochondrial genome or mitogenome, as I'm going to be referring to them. And these are also very useful for explaining evolutionary relationships, and they are useful for population studies. And just briefly, the bioinformatics part, for the UCE, I use a program called phylogeography, which is specifically designed to work with this data. And it's just a bunch of functions and steps in order to get your final alignments. When you get raw reads, you clean your data, you assembly, and then you have your final alignments. And for my mitochondrial genome, what I did was using 2 different tools that basically you can take one genome as a references, and then you can find parts of that mitogenome in your samples. Then you build a consensus mitogenome, and then you have your assemblies, and then I finally got my final alignments. And this is my final data set. And from this, all I would like you to pay attention is that these are big data sets The base pair number is how wide, like how long the alignment is. So the UCE, since the nuclear gene it's longer, we have 1,000 million of base pairs, whereas in mitochondrial genome we have only 15,000 base pairs. So now I am ready. I have all my data sets. Now I can start doing other fun things. The first thing that I did was population genomic structure. I work with single nucleotide polymorphisms, or SNPs, which are variations at a single position in a DNA, and these are very useful to characterize genetic diversity in populations. I use a discriminant analysis of principal components in order to find genetic clusters based on the shared SNPs. And this is just an example of what it is. You get some clusters here in the genetic space, and then you can plot this on a map so you can try to see if there's some like correspondence with the geographic location of these clusters. So I did that for all of my species. I'm not going to be explaining that because I'm going to bring this later. Then, so I perform my phylogenetic analysis. For this part, I perform maximum likelihood trees in both, in per species and for both datasets, nuclear and mitochondrial DNA. I used IQ3 and I run 10,000 ultra-fast bootstrap replicates for no support. And then once I did that, I had phylogenetic trees for all of my species for both datasets. So now I was done with my evolutionary histories, and now I can do my time-calibrated phylogenies. So once I have my phylogeny, you can actually put some dates. We have like the topologies, we have events here, and then we can estimate some dates here, and then that will give you more idea of the historical biogeography of these groups. So for doing that, I use a method called relative time, which is a fast dating but a high performance method that allows you to work with large genomic data sets like the ones that I was working. And it works using some calibration constraints. Basically, you have to know some divergence times, like prior knowledge of some divergence times. So in order to build these time-calibrated phylogenies, I first perform an additional like super tree where I did one single alignment with all of my species, and I included a lot of outgroups. And then this is one example of the calibrations constraints that I used. The Colostetus genus is the sister genus of the America. So based on past literature, we have an idea of when these two genus diverge. So we can use this as a calibration constraint to inform the tree and then estimate the other times. So now we have the time-calibrated phylogeny, evolutionary histories. Now I can start doing the last part, which is the historical biogeography. So for that, I first perform an ancestral range estimation analysis. What it means is that, well, once you have your time-calibrated phylogeny, so this is very useful for this analysis. And then if you have knowledge of the current ranges of these like tips of the tree of the samples that you have, you can estimate the ancestral ranges of the nodes that are in like more intern in the tree. So to do that, I use a program, an R package called BioGeoBears, which uses a probabilistic inference through a maximum likelihood algorithm to estimate those ancestral ranges. So this method works with predefined biogeographic regions. So I use these bioregions. I'm going to be referring to them as bioregions that were previously estimated by other researchers. These are regions of unique Anuran species composition, and they determine that there are 8 regions in the Amazon. The ones that are in gray, it's because I don't have samples there, so I'm going to be focusing only on the ones that I have colors. And once I did this analysis, the next step was to run a biogeographical stochastic mapping, or BSM. And what this analysis does is that it runs several simulations of these other previous analyses. And then once you have a lot of simulations, you can estimate directionality and also frequency of migrations through time, and that helps you to understand the biogeographical histories. So now so I performed that analysis for all of my species and my data sets. However, today, because of time constraints, I'm just gonna be talking about the nuclear DNA. So now I have all of my steps, evolutionary histories, time-calibrated phylogenies, biogeographical histories. Now I'm gonna start telling you my results. But before I do that, I need to explain you a little bit about this figure because you're gonna see it a lot throughout this talk, and I just wanna make sure that you understand what I'm saying. So here are all my results together. We have in here we have the phylogeny. You can see that it's calibrated at the bottom. They're like million years ago. The time goes from the past to the present. And then you see a lot of colors in the tree. So the colors match the colors in the map. The squares in the tips of the tree represent the current ranges of all of the samples that I have. And then the pie charts in the tree represent the actual ancestral range estimation of that analysis. So I'm going to be also mentioning some concepts. For example, whenever I talk about expansion, that means that there was movement within the same area or bioregion. For example, here we see that the ancestor right here was estimated to be in the upper Amazon, and then from there it just moved within the same area. However, if I talk about dispersion, that means that one ancestor that was estimated somewhere else, like here in the upper Amazon bioregion here, it moved or it migrated into a different area, like this one. So this is a dispersal event. And finally, we have vicariance events, which what that means is the splitting of an ancestral range. So basically, here we have one ancestral range. Sometimes the estimated ancestral range are not going to be completely corresponding to the colors in the map, because you can have ancestral ranges that are combinations of other areas. So whenever that happens, I'm just going to explain it. So here we have that the ancestral range of these in populations was estimated to be in several bioregions. However, whenever there was a split, we have one group that stayed in this area, in this pink or orange, I guess it's pink, and the other one that stayed in this other area, the Central Amazonia and Brazilian Shield. So this is a vicariant event because you're splitting the range. So now now that I think that I explained that better, now I'm gonna start talking about my results. So first I'm gonna talk about Amerega trivitata. So for this species, this is the phylogeny. As you can see, it's calibrated, and we recover high supported phylogenies, where I detected 2 early divergent clades right here. And the crown age of this species, meaning that is when the sample population started diverging, it was estimated to be 2 million years ago. And then from there was a split into 2 different clades. The clade number 1 right here started diverging 1.6 million years ago, and the other clade did it a little bit earlier, 1.9 million years ago. So now, when we talk about the ancestral range estimation, what we obtained was the ancestor was estimated to be widely distributed in all of these bioregions: Upper Amazon, Central Amazonia, and Brazilian Shield. And then after that, like widely distributed ancestor, we got a vicariance event that split populations right here in the Central Amazonia bioregion and Brazilian Shield from the rest of populations. So as you can see, these populations are now completely like differentiated from these populations that has an ancestral distribution in the Upper Amazon bioregion. So after that, this clade number 2, the one with ancestral distribution here, it experienced some expansion within this same bioregion, and it also some dispersion into the Juruapurus bioregion and also into the Central Amazonia bioregion. And my BSM results, basically, they confirm these migration paths. As we can see here, strong migration detected into this area, but also migration from here the upper Amazon into the central and also into the Brazilian Shield. So that's for Amerega trivitata. Now for the other species, Amerega haneli, this is the phylogeny that we obtained. Again, we recovered two clades. The crown age of this species was 3 million years ago, and then after that we have an split where clade number 1 started diverging 2.3 million years ago, and clade number 2 right here started diverging 2.5 million years ago. Now, in terms of their ancestral ranges, what we obtained was this ancestor was also widely distributed. As you can see, it shared the same two upper Amazon and central Amazonia bioregion. However, this ancestor was also estimated to be here in this region, the Juruapurus bioregion. After this widely distributed ancestor, we detected a Vicarian event as well, where populations here in the upper Amazon bioregion, these were restricted here to this area. And this happened like around 3 million years ago, and then the other population, the other clade maintained or the ancestral range right here. After this, we detected another Vicarian event here that splitted populations from this bioregion, the upper Juruapurus, with the rest of them. The other ancestor here, one went here to the Juruapurus, and the other one was estimated to be around the central Amazonia bioregion, as you can see here. So this ancestor then later experienced some expansion within the same bioregion and then into the Brazilian Shield. And the other thing that happened with this ancestor is that it dispersed back to the west, first into the Juruapurus bioregion and then into the upper Amazon. And when we analyzed this with BSM, we confirmed these results. We detected migration here and also this migration route back to the west. And then finally, for my Ranitomea variabilis amazonica species complex, this is the tree that I got, where you can see that the divergence time between variabilis and amazonica was estimated to be 3.3 million years ago. And then after that, the Ranitomea variabilis species started diversifying 2.9 million years ago, whereas Ranitomea amazonica did it a little bit earlier, 2.2 million years ago. In terms of their biogeographical history, the ancestor of this species was estimated to be in this region. It was a more restricted ancestor. After that, we have the divergence between these two species, and then within the variabilis clade species, what we got is, like, you can see that these populations right here, they diverged very early on time, around 3 million years ago, and they just appear here. So this is a way long dispersal, and this is what we call jump dispersal. After that, the variabilis only expanded its distribution along the same bioregion, and then the amazonica, the Ranitomea amazonica species, it also expanded its distribution, but also dispersed into the central Amazonia bioregion. So now that we have, and now we have the BSM analysis that we recovered this migration here, and we have some like migration arrows from the west into the east, and also within these two bioregions, the Eastern Guiana Shield and the Brazilian Shield. However, if you notice the numbers, like, there's not a lot of resolution or conclusive results. And whenever this is nuclear DNA, but whenever we analyze the mitochondrial DNA, we actually get a little bit more resolution, and then I'm going to be talking about it later. So that's my historical biogeography part, was basically trying to understand how these species have migrated along this area. So now the next part of my talk will be the comparative phylogeography part, which it's intended to try to summarize this and try to find spatial and temporary congruences in the biogeographic histories of these widely distributed poison frogs. So if we want to, okay, first, something that I got with my results was that in all species, we obtained that an ancestral lineage split very early in time into two different clades. This is very interesting. And also because one of the clades was always restricted and the other clade is the one who colonized other areas. However, the processes responsible for this divergence were different. As if you recall, we detected vicariance events in these two species, and this is according with the fact that they had widely distributed ancestors, and then something happened, and then their ranges were splitted. However, for Ranitomea variabilis, what we recovered was a very restricted ancestor that made all the way to the east. Also, well, all of these species have eastern populations. However, the timing of where these populations were established was different. For example, for these two Amarega species, it was during the Pleistocene, like 2.5 million years ago from now. And then for Ranitomea variabilis was late Pliocene, so a little bit earlier than the other one. With Trivitara, it's a little bit different because Trivitara was already estimated to be there by the Pleistocene. So now that we kind of know a little bit more about what happened with these species, now I want to understand what are the processes that drove those mechanisms. So if we want to understand that, and we're talking about Amazonian frogs, we need to talk about the Amazonian historical dynamics. And so in order to understand how these dynamics have influenced the phylogeographic and biogeographic patterns of widely distributed species. So here I am showing you a picture of the Amazonia, how it looks right now in the present time, where you can see here that there is this is the Amazon River, and then you see like the tributaries right here, and we see a huge river network. We see here in purple-ish, these are the Andes. However, the Amazon, as we know it right now, it wasn't always like that. And this region has experienced a lot of changes that date back to the Miocene, and these changes are due to geological activity, fluctuating drainage and hydrological patterns, and also climate change. So I'm gonna just talk a little bit about this history because it is important for our species. So before, like long time ago, the Amazon River didn't exist. As you can see in this image, there were some rivers flowing here, but we don't have a river that is like flowing to the east as we know right now. Instead, we had a huge lake right here in Western Amazonia that was a combination of different processes, like here in red, what you are seeing is the Andes that are uplifting. We don't have the Andes mountain range yet completely formed. So this mountain range was still lifting. Then we had some marine incursions. So there was a lot of water retention here in this area. And this is what we call the PIVA system, which was like a lake system. With time, and with the continuous erosive activity of the Andes uplifting, and also like erosive activity, sediment deposition, this system that was once a lake, it changed. So the landscape changed from a lacustrine system into an eastward flowing fluvial system. So now we start seeing that now the water is flowing to the Atlantic. So we could think about this acre system as an Amazon precursor. Then, after we started having this like flow of water, the wetlands that were here, they started to drain. This was a very slow process. They started draining very like progressively, and then this is when the courses of the Amazon rivers just started to to form, and the rivers started to become a little bit more defined. During this process, there was a conversion of the landscape here, because as the rivers crave more and deeper and permanent channels, then the forest right here also changed from flooded habitat into terra firme forest. So now that we know a little bit about our biogeographical histories, and now we know also a little bit about Amazonian history, now I can start to formulate hypotheses to try to understand how these processes were influencing the biogeographical histories of Mayan species. So first of all, if you remember, we recovered that in all of the species at some level, this region right here, the upper Amazon bioregion in western Amazonas, it was estimated as an ancestor of all of my species. So what we think that happened is that this huge wetland system that was there for a long time, it prevented dispersals towards the east. And this is consistent with the idea that all poison frogs originated in the east, and then it was just after there was a recession of all of these wetlands, they started migrating. And these two genera actually also originated in the western Amazonias. And this is a pattern that is also recovered in passerine and non-passerine birds, where around that time there were more stable populations here in northern and western Amazonia. However, for these two species, the ancestor was not only here, but also here in other regions. They were more distributed. So we think that what could have happened is that if there were some dispersals, they were mainly restricted to the south, because there was a huge water system here. Then, so at the time that these rivers were forming and there were a lot of changes, the Andes activity continued, and there was a massive wetland also in this area. And we think that this process could be the responsible for this Vicarian event that we have right here, because of the geographic and also the timing of these events. With this clade, the early divergent clades, there's something interesting that we found, and is that when you analyze the nuclear DNA and the mitochondrial DNA here in this side, I guess you're right, we recovered the same two highly supported clades in both of the phylogenies. We also recovered that these populations right here, this clade was also recovered as a one unique population right here. So what this might tell us is that this clade, or this species in general, needs some taxonomic evaluation, because this could be a potential candidate species. Of course, in order to confirm that, we need to do some analysis like call and morphology and niche divergence in order to be able to confirm this data. However, what it is important, at least for me, is that even though if we find out that these are two different species, we will still recover one widely distributed lineage. So with that, which is also interesting in terms of this dynamism. So moving on, we think that these wetlands around this time in the late Pliocene could explain the divergence that we obtained here within these two species, because they were here and then they got split here. So we think that this might explain this divergence. Now we're gonna focus on the Ranitomea variabilis species right here. So I'm gonna explain you what are the hypotheses that we have for this jump dispersal event that we detected right here. We know that it was a long time ago during this highly dynamism, where the rivers were still forming. So I'm going to introduce you to this dispersal hypothesis that is called rafting. Rafting is a passive dispersal mechanism that it is used to explain long distance dispersals across rivers, and across oceanic barriers. And what it basically says is that individuals they raft on floating debris, and that's the way they are able to get into new ecosystems. And this is very studied in islands. There are a lot of studies that show that. And this is just a picture from a explaining one paper, a very recent paper, where they detected that this was the mechanism of how some iguanas from North America made its way into Fiji. Just like they traveled 8,000 kilometers just to get there using this mechanism. So along the Amazon River, there are several ways that could facilitate this form of dispersal. For example, we have macrophytes. There are floating meadows in the rivers, and there are also something called vegetative rafts, which basically they are like chunks of vegetation that they are from landslides, and they carry trees and all of what a tree can have in that, like animals and plants and all of that. And these are actually very frequent and very common to observe in this region during the rainy season, and also due to tectonic activity. So what we think that could happen in that time is that there was a highly dynamic river, and then somehow this species, some individuals of this little tiny frog, they just rafted to the other side. And we have some other reasons to think that this might be the most likely mechanism, mainly because we don't think that terrestrial dispersal is actually possible, because this is a very tiny frog, and it also has a territorial behavior in which they basically have to stay in one small territory, so they don't disperse a lot. And the other thing about their behavior is their reproductive behavior. So this species is highly associated with bromeliads. So the females lay eggs on bromeliads. The males deposit tadpoles in bromeliads. So they basically spend their lives in bromeliads. So we think that what could have happened, all of these, even though they are against terrestrial dispersal, they actually could favor this rafting idea. And so what we think is that somehow bromeliads that had tadpoles for adults, they just made all the way to the east. And there are some studies about these rafts on rivers. And the thing is that there are a lot of things that can live in these rafts. There are insects, there are like a lot of diversity there. So we could think that they were probably not having a really bad time. They had food, and they were like just enjoying the ride. So here with this is where I'm gonna bring up the mitogenome for this talk, because we discovered something very interesting in this species. There's a long history of introgression in a contact zone. So in this blue circle, this is a contact zone, which means that in that area, both species, they coexist. So what we detected is that this is the nuclear DNA, and this is the mitochondrial DNA. Here in this part in gray, we have Ranitomea variabilis, and here we have Ranitomea amazonica. So what we detected is that this clay right here, which corresponds to the eastern populations, if you follow these lines here, their mitogenome is nested within Ranitomea amazonica samples. So what that means is that the individuals that made all the way to the east, they were probably having fun here, and they captured the mitogenome of Ranitomea amazonica here. And this is one of the reasons of why these two species have caused some headaches in terms of their taxonomy. Because if you study the previous studies, they used 16S gene, and they detected that they were part of this group, the Ranitomea amazonica. However, when they used Jason's previous student, she used UCEs, and she actually transferred those populations here. So now we know that it's not only one little part of the mitochondrial genes, it's the entire mitogenome that it was captured by this species. And then the other fun thing is that we detected that these populations right here belonging to Ranitomea amazonica, as you can see right here, they also have mitogenomes that are associated with Ranitomea variabilis. So we detected this mitogenome capture in both ways. Well, now moving on from this history of these Ranitomeas, I'm just bringing this again because I just want you to remember that there was one time when there was a wetland here, and then there was a conversion from flooded forest into terra firme forest. So what we think is that this flooded forest might have partially limit the dispersal of these Amarega species because they are not associated with water, with these ecosystems. They are only usually found in terra firme habitats. And this is however, this is a very big frog. As I said before, for being a poison frog, is a big frog. And so it also helps the dispersal. It actually travels a lot. So these species, as many other poison frogs, they transport tadpoles into pools. So what they do is that when they travel, males, they travel hundreds of kilometers away in order to deposit these tadpoles. Meters, hundreds of meters, not kilometers. I mean, they're great, but not that great. So they travel a lot. So we can think about these floated systems like just a mosaic of wetlands that they might serve as temporary pools, and they will just like gradually dispersing into the landscape. However, with these other species, Ranitomea hanley, the story is a little bit different because we think that these species disperse through like following this landscape conversion. And the reasons why we think that is because this species is highly associated with these habitats. She doesn't care if it's floated or not. She's all around here, and it's actually naturally dispersed through Barcia and through this ecosystem. So now I'm going to show you a picture. I have to explain it before because it's not going to be very like clear. So I'm going to show you a picture of Amarega hanley individuals that are riding on a slot, and I'm going to explain this story right now. So this is a documentation of an event where some researchers found a dead sloth that was floating on a river. The sloth was probably killed by a jaguar, and they realized that there were a lot of frogs here having fun, eating from all of the ants and other insects that were there. And what is very interesting is that this sloth was found 18 kilometers away from the nearest terra firme forest. So they can actually disperse through this habitat using many different rights. So, and this is important also because we detected some evidence of rafting here in this group too, in this species too, because this sample right here was nested within these other, like more western populations right here. So there might be a reason to think that these populations right here also made the way all the way to the east, just using a ride to get there. So with that, I finished. So I'm just going to go very quickly with my conclusions. So I uncover complex evolutionary histories and biogeographic histories. I detected ancestral distributions in Western Amazonia with migration in and out of it. In all of these species, we had two deeply divergent clades that with one was restricted and the other one expanded its distribution. We detected vicariant events in all species, however, different mechanisms. Then we have rafting events for at least 2 species. However, this needs to be like further investigated. And then in terms of the phylogenetics, I have some new discoveries. For example, the clade in Amarega hanley that needs to be evaluated as a potential candidate species. And my results on this Variabilis amazonica species complex, they provide some clarity to the taxonomic inconsistencies that we had in the past. since we detected this historical contact and introgression. We detected some discordance between the mitochondrial and nuclear DNA, which demonstrates the need to perform analysis on both of them before we can make any inferences in taxonomic or biogeographic analysis. Okay, so that's my conclusions. Now I'm just gonna quickly talk about future directions of this project. I would like to do some, like we have now some hypotheses to think about what happened. So I would like to do some hypothesis testing, some more like quantitative testing, and where I want to include and explore the impact of climate change. We know that there has been a lot of climatic variation throughout the Pleistocene. So, and at the end, as in several other aspects in biology, one mechanism can probably not explain everything. So there's probably a synergy of several processes. And as we see before, each, like the processes can affect taxa in different ways, depending on life history, behavior, dispersal ability, and physiology of the individuals. So now I have to start with my acknowledgement parts, and I promise I won't cry, I will try. But first of all, I want to say thank you to all of my committee members, Dr. Frank Anderson and Kurt Newbeck, for all of their support, and of course my advisor, Jason, which of course deserves a special mention. Jason has been, oh my gosh. So Jason has been a very kind person. He's been very understanding in every matter. He was very welcoming since the first moment when I contacted him, and I don't know how he answered my email, but he did it, and here I am. So I'm happy about it. I'm happy you saw the come here, and well, it's like a process, like a roller coaster, and he's always and it's not only Jason, but also his family, Jen and Macy, because they open the doors of their house for us, and they have been really amazing, making me and us, like other international students, oh my gosh, I'm gonna cry now, making us feel like we have like a family here, and that we are welcome. So these are some pictures of us in the field, just getting tired and needing some little help from people of the region, because it was really hard. Then I have to say thank you to my lab members, my lab mates. The past members, you might recognize Aleida here and Jacob, and these are, so I want to thank my previous and current lab members, because they have been, okay, they have been amazing. They have helped me a lot to go through this process, okay. And the thing with them is that they're not only okay, this is the lab, as I call it, like sensu stricto. This is the formal members of the lab, and this is the sensu lato, where of course Chris, Taylor, and Lady are there, because for me we are all the same thing, like. And the thing with these lab mates is that they're not only my lab mates, they're also my friends. We've shared so many moments together, and I would not be able to make it through this point if it wasn't because of the help of all of them, and also other friends that I've made here, all the grad student community right here, which I don't have pictures of all of us, but I really appreciate all of your help and everything. Oh my gosh. So for me, being here is not only about my master's, it's about, as Jason mentioned before, it's about all of my journey coming here, which is it was a very hard process at the beginning. However, I've enjoyed a lot, and I've made a lot of friends here, who all of them are in the, like, not all of them, because a lot of people is gone, which is what happened when you just come to study, no matter where you are from. You just leave, but all of them have been very important in my growth here as a person and as a scientist too. And these are some of my Carbondale family, and I definitely have to say thank you to my family. This is my small family, parents, siblings, and sisters and nieces, and my dog here, of course. I wouldn't be able to be here if it wasn't for them. So to all of them, I want to say gracias porque ustedes me han apoyado todo el tiempo desde que decidí venir acá y a pesar de la distancia han estado ahí siempre, siempre, siempre apoyando. Entonces, pues muchas gracias por eso. And I have a huge family. I have cousins, I've had grandparents, I have aunts, and all of them are very important for me, and they have helped me a lot to go through all of this process. So, gracias a mis tías, a mis tíos, a mis primas, porque también han sido gran parte de este viaje. And as I was looking for pictures with my family, I realized that this has been my life in the last 4 years, just me trying to keep connections with them through phone calls. And it has worked, I guess, till now, even though I'm not very responsive sometimes. And finally, these are all of my other friends who are not here. They are either in Costa Rica or in other places doing her projects. And all of them have also been very important in this process because they know me and they know a lot of things about me. And every time I go there, we just have an amazing time. And then I came here and then I disappear. And then I go back and then it's like nothing happened. Okay, so with that, I just want to say thank you, gracias a todos, and if I might have time for questions, if you have.